Good morning, everyone. My name is Kunwar, and I am from MSc Biotechnology, 12th third semester, and my roll number is 90LS301022. And our faculty name is Mr. Manikandan sir. So today I will be talking about gel electrophoresis, in which I will talk about PACE and PFG. So introduction. Electrophoresis is a separation technique which is based on a movement of charged particles in an electric field. Gel electrophoresis is a method for separation and analysis of micromolecules like DNA, RNA, and protein and their fragments based on their size and charge. So polyacromate gel electrophoresis is a method to separate, identify, and purify biopolymers. Since both these gels are porous in nature, polyacromate gels are chemically cross-linked gel, which is formed by polymerization of acrylamide and bisacrylamide. Gels are made by free radical induced polymerization by acrylamide tamide as the catalyst. Acrylamide gel electrophoresis is a technique which is widely used all over the world. The most commonly used form of polyacromate gel electrophoresis is SDS space, which is used mostly for the separation of proteins. Talk about principle of PACE. Principle SDS is an analytical method used to separate components of a protein mixture based on their size. The technique is based upon the principle that a charged molecule will migrate in an electric field towards an electrode with opposite sign. The general electrophoresis technique cannot be used to determine the molecular weight of biological molecules because the mobility of a substance in the gel depends on both charge and size. In SDS space, the use of SDS and polyacromate gels are largely eliminates the influence of structure and charge, and proteins are separated solely based on polypeptide chain length. Towards procedure, first we will have to prepare a prepare sample in which samples may be any material containing proteins or nucleic acid. The sample to analyze is optionally mixed with chemical denaturant if so desired. SDS is an anionic detergent that denature secondary and non-disulfide linked tertiary structure and additionally applies a negative charge to each protein in proportion to its mass. Then urea breaks the hydrogen bonds between the base pairs of nucleic acid, causing the constituent strands to anneal. Heating the samples at least 60 degrees Celsius further promotes denaturation. So, and tracking the dye may be added to the solution. Then preparation of gel. The gel typically consists of acrylamide, bisacrylamide, and buffer with an adjusted pH. The ratio of bisacrylamide to acrylamide can be varied for special purposes, but it is generally about 1 part in 35. Lower percentage gels are better for resolving very high molecular weight molecules, while much higher percentage of acrylamide are needed to resolve smaller proteins. Gels are usually polymerized between two glass plates in a gel caster with a comb inserted at the top create the sample walls. After the gel is polymerized, the comb can be removed and the gel is ready for electrophoresis. Then we will do electrophoresis, in which various buffer systems are used in PACE depending on the nature of the sample and experimental objective. The buffer used to use at the anode and cathode may be different or same. An electric field is applied across the gel, causing negatively charged protein or nucleic acid to migrate across the gel away from the negative and towards the positive electrode. Depending on their size, each molecule moves differently through the gel matrix. Small molecules more easily fit through the pores in the gel, while larger molecules have more difficulty. The gel is run usually for few hours, though this depends on the voltage applied across the gel. After the set amount of time, the biomolecules will have migrated a different distance based on their size. Smaller biomolecules will travel farther down the gel, while the larger ones remain closer to the point of origin. Then visualization. After the electrophoresis is completed, the molecules in the gel can be stained to make them visible. Then ethidium bromide, silver, or Comesi blue dye can be used. Other methods can also be used to visualize the separation of the mixed component gel. After staining, different species biomolecules appears as a distinct band within the gel. Then some application of PACE. It is used for estimation of molecular weight or proteins, and nucleic acid. It is used for determination of subunit of structural protein, for purification of isolated protein, for monitoring changes of protein contained in body fluid, and comparison of polypeptide composition of different samples. Then talk about PFG, which is also which is known as pulse width gel electrophoresis. It is a technique used for the separation of large DNA molecules. As DNA larger than 15 to 20 kb migrate through a gel, essentially moves together in a size independent manner. This standard gel electrophoresis technique was unable to separate very large molecules of DNA effectively, which led to the practice of pulse width gel electrophoresis. In 1982, Swords introduced the concept that DNA molecules larger than 50 kb can be separated by using alternating electric fields principle. While in general, small fragments can find their way through the gel electrode more easily than large DNA fragments. A threshold length exit about 30 to 50 kb, where all the large fragments will run at the same rate and appears in a gel as a single large diffuse bands. However, with periodic changes in field direction, the various length of DNA react to the changes at the different rates. So there are some steps involved in PFG, which is like lysis, then the digestion of DNA, then electrophoresis, then analysis. So talk about lysis. First, the bacterial suspension is loaded into agarose suspension. Well, okay, here. So here you can see as there is a bacterial culture in which where we will add a buffer to make a suspension, then we will add a molten agarose to make a plug, then agarose plug will fill with bacteria, then we will lysis the cell, then where the whole genomic DNA will be made, and we will digest the enzyme where DNA fragments will be isolated through electrophoresis. Talk about application of PFG. Since filled gel electrophoresis allows the separation of DNA fragment containing 100 kilobase pair, the characterization of such fragments has allowed construction of physical map for the chromosome from several bacterial species. PFG may be used for genotyping or genetic fingerprinting. It is commonly considered as a gold standard in epidemiological studies of pathogenic organisms. There are some advantages of PFG as it separates DNA from few kb to over 10 MB pairs. PFG subtyping has successfully applied to the subtyping of many pathogenic bacteria and has high concordance with epidemiological relatedness. It has been repeatedly shown to more discriminating than methods such as ribotyping or multi-locus sequence typing for many bacteria. PFG in the same basic format can be applied as a universal genetic method for subtyping of bacteria. DNA restriction generated by PFG are stable and reproducible. These are my references. Thank you.